Hello. We are going to begin today by discussing the process of meiosis, and you've probably already learned about the process of mitosis, and meiosis has some similarities. So there are also some important differences. So we like to call meiosis the reduction division because instead of giving us the same number of chromosomes at the end, we end up with half the number of chromosomes. So similar to mitosis, in meiosis, beforehand, when the cells are getting ready, the chromosomes are duplicated. But what ends up happening in meiosis is that there's two consecutive cell divisions, meiosis 1 and meiosis 2. And at the end, when you finish meiosis 2, you're going to end up with 4 daughter cells, and each of those cells will only have half as many chromosomes as the parent cell. Meiosis takes place in order to create gametes, which are either your sperm or egg cells, and those are the cells that are going to be responsible for sexual reproduction. So that is the only type of cell where meiosis will occur. In meiosis, there is a single pair of homologous chromosomes in a diploid cell, and both of those get duplicated. And when we talk about homologous chromosomes, typically you'll see them as two X's hanging out. And these homologs have different versions of alleles, or different versions of the gene called alleles, but they do code for the same gene. So these homologous chromosomes hang out together, especially during meiosis, but otherwise they don't have any obvious association with each other. So in our meiosis division, this is just a very simplified version showing you what happens with our chromosomes. So in interphase, before the chromosomes get duplicated, we have just one of each, but then they are duplicated in interphase, and we end up with what are known as sister chromatids, and those are the two duplicated chromosomes that are exactly the same. They hang out together like this. However, there is another chromosome that codes for the same gene but may have different versions of that gene known as alleles. And these two together make up a homologous pair, sometimes referred to as homologous chromosomes or homologs. Like I said, they code for the same gene, but they might have different alleles. Then in meiosis 1, these homologous chromosomes separate. So now you have one of the homologous pairs members over here and the other member over here. In meiosis 2, then, we get a second division, and now the sister chromatids separate, and we end up with 4 daughter cells, which are haploid, because notice originally we had 2 chromosomes in the original parent cell. Now we have 1 chromosome in each one. So our daughter cells are now haploid, and we end up with 4. So essentially what happens in meiosis is that the total number of chromosomes is halved, reducing the number of sets of chromosomes from 2 to 1, and each of the daughter cells receives 1 set of chromosomes. In the first part of meiosis, so known as meiosis 1, the homologous pairs of chromosomes separate, and in meiosis 2, the sister chromatids separate. So what you should keep in mind when you're trying to compare meiosis and mitosis is that meiosis 1 is really the one that's different. That's where you're just separating your 2 homologous pairs. But in meiosis 2, the sister chromatids separate, and overall, meiosis 2 looks very similar to mitosis. And then in the end, you get 4 new haploid cells, each of which is genetically unique. So those 4 cells are different genetically than the parent cells. Whereas in mitosis, remember, you got 2 genetically identical cells. That's another difference. This kind of shows you in picture form what's happening here in meiosis 1. Notice we have our homologous pairs hanging out. Then they line up in the center in metaphase. those homologous pairs get separated, and after telophase 1, you end up with one of the homologous pairs in every new nuclei. So you get 2 new nuclei, each of which contains one of the pairs. So over here you notice we had a blue and a red, and we had 3 different pairs, so a total of 6 chromosomes. Now we have different ratios, but we have 3 chromosomes in each of the nuclei. Now, moving on to meiosis 2, you're going to see this is the one that looks a lot like mitosis, because those pairs, these chromosomes are now separated from their homologous pairs. They're now single chromosomes, but they still are composed of 2 sister chromatids held together by a centromere. They're going to line up in the middle in metaphase, they're going to separate in anaphase, and we end up with our haploid daughter cells. So if we go back to our first page where we started in meiosis 1, or here's a little bit bigger, we had 1, 2, 3, 4, 5, 6 total chromosomes. Now, at the end of meiosis 2, we have a total of 1, 2, 3, 1, 2, 3 in every cell, and they're no longer pairs. Also, pay attention to the genetic makeup of these chromosomes. You're going to notice if you go back again to meiosis 1, they look very different. So one of the most important stages in meiosis is prophase 1. Similar to in prophase and mitosis, your chromosomes condense, but what's also important in meiosis is that the homologous chromosomes pair up, and they align themselves based on the genes that they code for. Then, once your chromosomes are paired up with their homologous buddies, a process called crossing over occurs. And this is where non-sister chromatids exchange DNA. With our homologous chromosomes, each of these pairs has some X-shaped region, at least one, sometimes more, called chiasmata. And these chiasmata exist where crossing over has occurred. So if we take a look over in meiosis 1, you can see these chromosomes, these homologous pairs, are crossing over. It's almost like they hug and they switch feet, is the simplest way to look at it. But in doing so, that helps increase the genetic variation, because you'll see once they separate, they have now gained a part of the opposite homologue, essentially. And then when we end up at the end of meiosis 2, we get very different chromosomes than we started with. In metaphase 1, the homologous pairs line up and separate, just as the microtubules are attached to the kinetochore and extend and attach to the chromosomes, so they do in meiosis 1 as well. In anaphase 1, those homologous pairs separate, and in telophase 1 in cytokinesis, we end up with for the first time, our haploid cells. However, in meiosis 1 at the end, though the cells are haploid, each chromosome still consists of 2 sister chromatids. So you still have 2 copies of each chromosome, the duplicated version. Then cytokinesis will occur and you end up with 2 haploid daughter cells. These cells are then, once again, divided using either a cleavage furrow or a cell plate, depending on if they're plant or animals. And since the chromosomes still consist of 2 sister chromatids, the duplicated versions, there's no second duplication. Meiosis 2 will just begin. And in meiosis 2, you end up with your 4 phases, and like I mentioned, this is the stage that's very similar to mitosis. We end up with our spindle forming, the chromosomes condense, they start to move toward the metaphasial plate. In metaphase, they're now arranged in the middle, and the kinetochores attached to the microtubules. In anaphase, those sister chromatids are pulled apart toward the opposite poles. And now in telophase 2, we get the reformation of the nuclei. The chromosomes begin decondensing, and at the end of meiosis, we end up with 4 daughter cells, each with a haploid set of unduplicated chromosomes. Once again, very important, each daughter cell is genetically distinct from the others and from the parent. So these cells are totally different from the parent cell that we started with. Okay. That leads us to the end of meiosis, but just as kind of a little bit more information about crossing over, once again, crossing over happens during prophase 1. How this works is basically two homologous chromosomes pair up next to each other lengthwise, and they kind of arrange themselves so that their alleles are matched up. And then through a process called synapsis, the DNA molecules of the maternal and paternal chromatids are broken at the exact matching point, and then the DNA breaks get closed so that the paternal piece joins to the maternal piece and vice versa. So you kind of saw in picture form what happens, but they basically have this moment where we get our chiasmata forming and those pieces cross over. So you end up with the chromosome pieces switching between the homologous pairs. And in general, what happens is that the same alleles will be transferred, but it will be from one chromosome to another. As long as there's no problems, it runs very smoothly. So we end up with our chromosome pairs switching, and we end up with that crossover. And that's going to be very important for our genetic variation. Just as kind of a review for you, because mitosis and meiosis are very similar, here are some differences and similarities between the two. So mitosis, you get the same number of chromosomes in the new cells, and the new cells are genetically identical to the parent cells. Whereas in meiosis, it's a reduction division. So instead of having the same number of chromosomes, you go from 2 sets, diploid, to 1 set, haploid. And the cells are now genetically different from each other and from the parent. Meiosis also includes 2 divisions as opposed to 1 after DNA replication. Mitosis only goes through 1 round of division. There's 3 events that are unique to meiosis, and those 3 events all occur in meiosis 1. So that's why I mentioned meiosis 1 is the most different. Meiosis 2, you'll notice, is very similar to mitosis. So in meiosis 1, we have synapsis and crossing over, which is that part where the homologous chromosomes connect with each other and exchange their genetic information. That increases genetic variation. Secondly, at metaphase, the homologous pairs line up along the center as opposed to just the sister chromatids. And then finally, in anaphase, the homologous pairs are separated. So the first round of meiosis is all about separating the homologous pairs. And then when we get to meiosis 2, we can separate the sister chromatids. In mitosis, those sister chromatids stay together until the end of metaphase. And in meiosis, they stay together until the end of anaphase 2. So they really are together for a major portion of the cell division. Here is kind of a list, if you like it all in list form, of some of the similarities and differences between mitosis over here in the orange and meiosis over here in the purple. And we'll stop there for the last section.